Welcome back to the new session. In this lecture, we are going to start a new chapter, that is anatomy of flowering plants. Now, according to the new NTA guidelines, from this chapter, anatomy of flowering plants, 50% of the topics have been deleted. Like tissues, meristematic tissue, parenchyma, collenchyma, sclerenchyma, xylem and phloem have been deleted. And again, the secondary growth topics, secondary growth in stem and secondary growth in root are also deleted. Now, what are there in this chapter for the coming NEET examinations means tissue systems are there, means epidermal tissue system, ground tissue system and vascular tissue systems are there. And anatomy of organs, anatomy of dicot root, monocot root, dicot stem, monocot stem, dicot leaf and monocot leaf. So these parts are there, that's it. So very limited content is there and the chapter is going to be easy because the difficult topics have been deleted. Right, now let's start the chapter, that is anatomy of flowering plants. Now when we see externally any plant or animal, so we can find out similarities, we can find out dissimilarities or variations. Study of external structure is called morphology. So in NCERT, the introductory para, they say that we can see similarities and variations. We can see similarities and variations in external structure. What is the external structure called? Morphology. In external morphology of all living organisms. The major living organisms are plants and animals. Like, what are the living organisms? They have given that they are plants and animals. So children, listen, what does this sentence in NCERT mean? They are telling that if you see the morphological features or external features, you can find some similarities, you can find some variations in plants and animals, right? And next sentence, they are telling that we can see similarities and variations. In internal structure also. External structure you can find similarities and dissimilarities. In internal structure also you find similarities and dissimilarities, right? Then study of internal structure is called as anatomy. So they are introducing us to the chapter study of internal structure of plants. If you take, it's called plant anatomy, or we can just say it is called anatomy. Now in this chapter we are going to see anatomy of angiosperms, flowering plants in detail, right? So then they will tell that in angiosperms, what are the two groups of plants in angiosperms? We have monocots as well as dicots. Now in angiosperms, among monocots and dicots, they have different anatomical structures. Means root has a different anatomy, stem has a different anatomy, leaf has a different anatomy. So among angiosperms, monocots and dicots have different anatomical structures. Right? They have different anatomical structures. Now coming to the plant, what is the basic unit of life? Cell is the basic unit of life. In plant, cell is the basic unit of life. In NCERT they have given, in plants, cell is a basic unit. Anything which is lesser than a cell is called non-living. So now cells, if they are grouped together, they make up tissues. So then they'll tell cells are grouped or organized to form tissues. Now what are the plant tissues? Simple tissue, complex tissue, like that. And cells are grouped together to form tissue. Tissues are grouped together to form organs. Tissues are grouped to form organs. Now what are the plant organs? Root is an organ, stem is an organ and leaf is an organ. Now In plants, cells are the basic units, and cells are grouped to make tissues, and tissues are grouped to make organs. Now when we talk about tissues, we can classify tissues in various ways. Now basing on the types of cells present in the tissue, we can divide them into simple tissue and complex tissue. Tissues are divided into two types on the basis of types of cells present. On the basis of types of cells present. Now, if it is made up of same type of cells, then that is going to be called as a simple tissue. And if it is made up of different types of cells, then that's called as a complex tissue. If it is made up of different types of cells, then what is that called, children? It's called complex tissue. This is one way. And coming to the other way of division of tissues. Tissues can also be divided on the basis of their structure and location. So tissues are also divided on the basis of structure and location. Now, into how many types you are going to divide the tissues basing on the type of structure and location? There are three types. Now, if we have to see what are the three types, means the one which covers the outer part is called epidermal tissue system, right? So, what's the first one? The first one is going to be epidermal tissue system. And the third one, where xylem and phloem, vascular tissues are there, is going to be vascular tissue system. Now, leaving this and this, rest all comes under ground tissue system. So, this is the third one, and the second one is going to be ground tissue system. Now, we are going to see each system in detail. We are going to divide the tissues into two types on the basis of types of cells. If they are made up of similar type of cells, then that's called simple tissue. If they are made up of different types of cells, that's called complex tissue. Now, for example, if I take all black color markers, then this is called simple tissue. If I take different types of markers, then it's going to be a complex tissue. This is one type of division. And the second type is we are going to divide the tissues on the basis of the structure and function. Then we have three types: epidermal, ground and vascular. Take a screenshot. Then we'll talk about epidermal tissue system in detail. Right. Now, coming to the first tissue system in NCERT, that is epidermal tissue system. Epidermal tissue system is the outermost covering of any organ, let it be root or stem or leaf. So it forms the outermost covering of any plant organ. And under epidermal tissue system, what are the topics we are going to study? Epidermal cells, of course, that's the first thing. And there are certain openings which are called as stomata, that's the second thing. And some projections will be there from the epidermal cells, which are called epidermal appendages. So under epidermal tissue system, we are going to see three things. What are they? Epidermal tissue system. Now, epidermal tissue system, it forms the outermost covering of all the plant organs. Covering of all plant organs. Epidermal tissue system includes or comprises of, I told, the first one is epidermal cells. Then the second one comes the opening, which is called as stomata. And the third one is epidermal appendages. Epidermal appendages include root hairs and trichomes. So these structures we are going to see under epidermal tissue system. Let's see one after the other one. The first one is epidermal cells. Now, epidermal cells, when we draw any epidermis, we see that the cells are elongated and they are parenchymatous cells. They are compactly packed without any spaces, right? And they are parenchymatous cells. Now, let us write down epidermal cells. First point, they are parenchymatous cells. And epidermis is generally single layered. It is single layer. And the cells are elongated. And they are compactly packed without spaces. The cells contain a thin cell wall and they contain small cytoplasm. Small amount of cytoplasm they have just inner to the wall. They have very little amount of cytoplasm inner to the wall. And the function of this epidermis or epidermal cells is protection. So the function will be protection. So under epidermal tissue system, we are telling they are parenchymatous cells. It is usually single layered and they are elongated, compactly packed cells without spaces. And the cell wall is thin and the cells contain small cytoplasm inner to the cell wall. Inner to the cell wall they have cytoplasm. And the function of this epidermis is protection. And this epidermis, in some parts, it is covered by a waxy layer called cuticle. The epidermis on the outer side is covered by a waxy layer called cuticle. Let's write down that. Epidermis is covered on outer side by a waxy layer. What is that waxy layer called? Cuticle. A waxy layer called cuticle. Cuticle is present in the aerial parts. It's absent in the underground parts. So we can say cuticle is absent for underground portion means root. Is absent for the roots and it's present in the aerial portions like stem and leaf, we can say. So this is about epidermal cells. Now let's talk about the second component. What is that? Stomata. Now what are these stomata? Stomata are openings. The pore is called stoma, right? Or stomatal aperture, we can say. And this stoma on either sides is covered by bean-shaped cells, which are called guard cells. In dicot, the guard cells are bean-shaped, whereas in monocots, the guard cells are dumbbell-shaped. So means I am drawing here a dicot stomata. So in dicot, so let me write down here, this is dicot. In dicot, what are these cells called? Guard cells. The guard cells in dicots are bean-shaped. Whereas if we draw monocot, in monocot, the guard cells are dumbbell-shaped. So this is one dumbbell-shaped guard cell. And let's draw the other dumbbell-shaped guard cell. This is the other dumbbell-shaped guard cell, right? And we'll extend it. This is one dumbbell-shaped guard cell, and this is the other dumbbell-shaped guard cell. And this is stoma here, and this is stoma here. The inner walls of the guard cell. So here it is guard cell, and this is also guard cell. This is dumbbell-shaped, and here it is bean-shaped. The inner walls of the guard cell towards the stomatal aperture are having thick walls, means this margins of the guard cells have thick walls, whereas the outer margins have thin walls. So here I am making them thick. So they are telling that the inner walls of the guard cells towards the stomatal aperture or stoma have thick walls and the outer walls are thin. So parallelly here also we have to make these walls thick. The inner margins toward the stomatal aperture are thick and the outer walls are thin. And actually epidermal cells will be colorless because their function is not photosynthesis, their function is protection. But guard cells are special epidermal cells which have chloroplast. So these are special epidermal cells. Why they are special? They have chloroplasts. Right? So they have chloroplasts. Guard cells are special epidermal cells with chloroplast. And these chloroplasts helps in opening and closing of the stomata, right? And some cells in the vicinity of the guard cells are specialized. They are parenchymatous cells and they convert themselves into a specialized type of cells which are called as subsidiary cells. So if we have to draw the subsidiary cells, so the cells in the vicinity of the guard cells, they get specialized. They get specialized, and such cells, what are they called? They are called subsidiary cells. So here I draw 1, 2, 3 subsidiary cells. And for monocots, these are the subsidiary cells. So what are we saying? Some cells in the vicinity of the guard cells, they modify themselves into special cells which are called as subsidiary cells. So these cells are called as subsidiary cells. Let's write down. So what are these cells called? Subsidiary cells. And peripheral to the subsidiary cells are normal epidermal cells. Now if you have to show the epidermal cells, yeah, we can draw. So this is an epidermal cell, another epidermal cell, and another epidermal cell. For monocot, the epidermal cells will be linear. So this is one cell like this, and here we can draw another cell. So this is one epidermal cell and this is one epidermal cell. So stomata is an aperture. This aperture helps in regulating the water loss by transpiration, and it helps in gases exchange. So stomata helps in regulating the water loss by transpiration, and it helps in gases exchange for photosynthesis and respiration. The stomata means stomatal aperture. On either sides, it's covered by a pair of cells which are called as guard cells. In dicot, the guard cells are bean shaped. In monocot, the guard cells are dumbbell shaped. The inner margins of the guard cells towards the stomatal aperture have thick walls. The outer margins have thin walls. Generally, epidermal cells don't have chloroplast, but guard cells will have chloroplast. And these chloroplasts help in opening and closing of the stomata. In some plants, the cells which are in the vicinity of the guard cells, they convert themselves into special cells which are called as subsidiary cells. So here I showed subsidiary cells, here also I showed subsidiary cells, and outside to the subsidiary cells are the epidermal cells. So one which is the subsidiary cell, the second one is the guard cell, and the third one, what is it called? Stomatal aperture. Stomatal aperture means stomatal pore. 1, 2, 3, all these three together will make up stomatal apparatus. They make up stomatal apparatus. And where do you find stomata? Stomata are found mostly in the leaves. Little number of stomata will be there in the stem also. Root doesn't have stomata. So where do we find stomata? Stomata are usually found in the leaves. Some stomata will be there on the stem also, but stomata are absent for roots. So children, we discussed about epidermal cells, that's the first part, and we discussed about stomata also. Take a screenshot. Then we'll talk about epidermal appendages. Right. Coming to the third one, that's epidermal appendages. Epidermal appendages for root are called as root hairs, and epidermal appendages of stem, that's called trichome. Now what are root hairs meant for and what are trichomes meant for? Let's discuss. Now function of root is absorption of water and minerals. So if the root hairs are more, the surface area will be more. More the surface area, more the absorption, we can say. So we're going to say that is root hair. Is one epidermal appendages found in root organ. Now second one is stem hairs, which are called as trichomes. And where do we find trichomes? In the stem. Now root hairs are projections of epidermal cells. If this is an epidermal cell, the epidermal cells project out to make an extension that becomes a root hair. So root hairs, I'm drawing, they are unicellular. Whereas trichomes, if we have to show, trichomes, we say they are multicellular. Means I'm going to divide this into many cells. So trichomes are multicellular. That's the first point. Root hairs are unicellular, whereas trichomes are multicellular. And root hairs help to increase the surface area of root. They increase the surface area of a root. That's a function, so that root can absorb more water and minerals. Whereas trichomes, they regulate or they prevent the water loss by transpiration. Trichomes, what do? They prevent or they regulate water loss by transpiration. And trichomes are multicellular. In some plants, trichomes can be soft, whereas in other plants, it can be stiff. In some plants, they can be hairy. In in some plants they can be smooth or glabrous. And in some plants, they can be secretory even. They can secrete gums and latex and all those things. So they can be either soft or stiff. They can be hairy or smooth. It's also called glabrous. Sometimes they're even secretory also. So the function of trichomes is to prevent the water loss by transpiration, whereas the function of root hairs is to increase the surface area so that the root can absorb more water and minerals. So with this, we are closing epidermal tissue system. Take a screenshot. Then we will talk about the second tissue system, which is called as ground tissue system. Right? So epidermal tissue system, which is the outer tissue system, this is done. Now let's go to the second tissue system, which is called as ground tissue system. In the previous one, I told like the outer one is epidermal, the inner one, where xylem and phloem are there, is called vascular. Now leaving these two, rest all is called as ground tissue system. So what can we write under the second one, which is called as ground tissue system? It's also called fundamental tissue system. This is PYQ, children. Fundamental tissue system is also called as, in once in need they ask, the answer should be ground tissue system. So ground tissue system or fundamental tissue system. Except epidermal and vascular, rest all comes under ground tissue system. Except epidermal and vascular, rest all comes under ground tissue system. Now, ground tissue system is the tissue system where we can find parenchyma, collenchyma, sclerenchyma. All simple tissues are found in ground tissue system. So, ground tissue system consists of all three types of simple tissues. What are all three types of simple tissues? So, one is parenchyma. Second one is collenchyma. And the third one is sclerenchyma. Where do we find parenchyma? Where do we find collenchyma? And where do we find sclerenchyma? So we are telling that the parenchyma is found in the cortex region, is found in the pericycle region, and it is found in the medulla or pith region. This is also called pith, and it is found in the medullary race. So we are telling that the ground tissue has all the three types of simple tissues, that parenchyma, collenchyma and sclerenchyma. Where is parenchyma found? Parenchyma is found in the cortex, pericycle. All these are the anatomical parts. When we see the root, we will see the parts in detail. Found in the cortex, pericycle, medulla or pith and medullary race. And where can we find collenchyma? Collenchyma is found in the hypodermis. It's found in the hypodermis of dicot stem metis. And where do we find sclerenchyma? Now, sclerenchyma is found in the hypodermis of monocot stem then. So we are telling that sclerenchyma is found in the hypodermis of monocot stem, right? And collenchyma is found in the hypodermis of dicot stem. And parenchyma is found in these four regions. So with this, we are going to close ground tissue system, which is also called fundamental tissue system. Ground tissue system means leaving epidermis and leaving vascular, rest all comes under ground tissue system. It's a tissue system where all the three types of simple tissues will be there. That's parenchyma, collenchyma and sclerenchyma. Parenchyma is found in so regions, collenchyma in this region, sclerenchyma in that region. Take a screenshot, then we'll go to the third one, the important one, that's vascular tissue system. Right, now what's the third one? That is vascular tissue system. Vascular tissue system, why is it also called as conducting tissue system? Xylem and phloem, they are called as vascular bundles. What do? They help in conduction of food, water, right? So that's why vascular tissue system is also called as conducting tissue system. Vascular tissue system. It is also called conducting tissue system. Vascular tissue system includes xylem and phloem. Now, since the xylem helps in conduction of water and minerals, whereas the phloem helps in conduction of food. Since it is helping in conduction, we also call vascular tissue system as conducting tissue system. Now, which type of vascular tissue system is found in root, we have to see, and which type of vascular tissue system is found in dicot stem, we have to see, and which type of vascular tissue system is found in monocot stem as well as dicot leaf and monocot leaf also. Dicot leaf and monocot leaf. All these three will have similar type of vascular tissue system. Now let us draw the stele. Stele is an innermost anatomical part which includes this outermost lining is called pericycle. What is this outermost lining called? The outermost lining is called pericycle. And what is the center part called? The center part is called medulla or pith. Then the middle region has a vascular bundles. Now coming to the root, in root the vascular bundles are on separate radii. So here is xylem, it is on different radius. Again we can find xylem here for dicot. Again we can find xylem here, and again we can find xylem here. Xylem is on different radius and phloem is on different radius. Here is phloem. Again here is phloem. Xylem on different radii, phloem on different radii. Then it is called as radial vascular bundle, right? So roots, which type of vascular bundle they have? Xylem on different radii, phloem on different radii. Xylem on different radii, phloem on different radii. So it belongs to radial type of vascular bundle. And there is no cambium separating xylem and phloem. There is nothing between xylem and phloem. There is no cambium. So if cambium is absent, then we call it as a closed vascular bundle. So the vascular bundles of root are radial and closed. Why is it radial? Because xylem and phloem are on different radii. Why is it closed? No cambium makes it closed. And so dicot root, monocot root have, what do they have? They have radial closed vascular bundles. Now, in an alternate manner, xylem, phloem, xylem, phloem, xylem, phloem. So they are alternate and they are radial and closed. In this, if we see, now this xylem is having narrow vessel. And as we move in, it is having wider vessel. This xylem is having narrow vessel. As we move in, it is having wider vessel. Narrow vessel. As we move in, it is having wider vessel. Now, what is this condition called? Protoxylem out, metaxylem in is called EXARC condition. The first formed primary xylem is called protoxylem. The later formed primary xylem is called metaxylem. Protoxylem has narrow vessels, metaxylem has wider vessels. If protoxylem is peripheral, and if the metaxylem is inside towards the pith, what is that condition called? The condition is called EXARC condition. Radial closed EXARC condition of vascular bundles are found in dicot root and monocot root also. I think you understood it. Now let's go to which type of vascular bundles are found in dicot stem. Now for dicot stem, similarly, we will draw a stele. As I told, this is the outermost region which is called as pericycle. In pericycle, we find vascular bundles. The vascular bundles in dicot stem are perfectly arranged like a ring. This is one vascular bundle. A ring-shaped arrangement of vascular bundles is characteristic feature for dicot stems only. So I am drawing the vascular bundles, ring-shaped arrangement of vascular bundles is a characteristic feature of dicot stem. Now always the xylem will be towards the pith and phloem is out of xylem. So means I am going to show xylem here and phloem here. Now this is xylem. All this is xylem. Xylem towards the inner side, phloem towards the outer side. Now phloem we are showing with green color, right? Yes. This is phloem. Xylem towards the inner side and phloem towards the outer side. And if you see here, xylem and phloem are on the same radii. They are together. They are together. Since xylem and phloem are on the same radii, so we call them as conjoint. Do you feel that xylem and phloem are joined? Yes, xylem and phloem are joined. So for xylem, I'll use this color. So this is xylem. Xylem, this is protoxylem, bigger out, smaller in, bigger out. Smaller xylem element in, bigger out. What is the smaller one called? Protoxylem. What is the bigger one called? Metaxylem. So protoxylem in, metaxylem out. So that condition is called as ENDARC condition. So which condition is this? The first condition we can write it is ENDARC condition. ENDARC condition of xylem. And since xylem and phloem are together on the same radii, it is called as they are joined together. So it's called conjoint. It's called conjoint. They are on the same radii. They are joined together. It is called conjoint. And xylem and phloem are together, or something is there between them, which is called as cambium. They have cambial cells in between xylem and phloem. So here in root we did not draw anything between them, but here we are telling there's cambium between xylem and phloem. So if cambium is there between xylem and phloem, we have to write it as open vascular bundle. So conjoint open, NCERT says conjoint open vascular bundles are found in dicot stem, and the condition of xylem is ENDARC condition. What do you mean by ENDARC condition? Proto in, meta out is called as ENDARC condition, right? Now coming to the remaining, like monocot stem, dicot leaf and monocot leaf, which type of vascular bundles do they have? Now again, like for monocot stem, the stele is not there, and in dicot leaf, monocot leaf also we cannot tell stele is there, we call it as a mesophyll. So let's just draw the vascular bundle and see. So in monocot stem, we draw the vascular bundle like this, and phloem will show it on the top. For monocot stem, the phloem is here, and the xylem is this way. Metaxylem should be out, right? And this is protoxylem. Protoxylem in. So protoxylem in, metaxylem out means this is also ENDARC condition. And xylem and phloem together. This is xylem, this is phloem together in one vascular bundle only. So if xylem and phloem are in the same vascular bundle, we should use a word called conjoint, right? And did I show any cambium between xylem and phloem here? No, I did not show. If cambium is absent, then it's going to be closed. So conjoint closed vascular bundles are found in monocot stem, dicot leaf and monocot leaf. Conjoint open. Why is it open? Because cambium is there. Where do you find it? In dicot stem. Radial, xylem on different radii, phloem on different radii. Xylem on different radii, phloem on different radii. It's called radial. No cambium between them. Closed. Protoxylem out, metaxylem in. XR condition is found in dicot and monocot root. So where is usually xylem present? Xylem is towards the inner side. And where is phloem? Phloem is on the outer side of xylem. With this, we finished vascular tissue system. There's a question on vascular tissue system in 2021 NEET. Just check the PYQs papers. So these things only they asked. Then we can start anatomy of dicot root. Take a screenshot fast. Now let's talk about anatomy of dicot root. Now when we are talking about a dicot root, we should take some plant as a reference. So they'll be taking sunflower plant as a reference plant to study dicot root. Dicot means they show secondary growth and they become woody, right? But here we are not talking about woody dicot root. We are talking about anatomy of primary root, means a normal root which did not start secondary growth yet. So through diagram itself we can understand so many points. Let's go. Anatomy of dicot root. So we are going to take sunflower plant as a reference plant to study dicot root. So we know in anatomy what are the three parts we see for root and stem means epidermis, cortex and stele. One by one we have to do. Now let's talk about the epidermis of dicot root. The epidermis of dicot root is called as epiblemma, and we already know that the epiblemma cells are elongated. They are elongated. It is single layered and they are projected out to make root hairs, and they don't have any spaces and they are parenchymatous cells. So this is about epidermis, which we already discussed. Root is an underground part, so it will not have cuticle. So epiblemma, what it is? The epidermis of root is called epiblemma. It consists of single layered parenchymatous cells which are tightly packed without spaces, no intercellular spaces, and the epidermal cells are parenchymatous cells, but they don't have chloroplast. No chloroplast, because their function is not to do photosynthesis, it's for protection. So no chloroplast, no stomata also, because their function is not to help in transpiration and gases exchange. No chloroplast, no stomata, and since it's an underground part, we can say there's no cuticle also. So it just has projections. Those projections are called root hairs. There will be millions of root hairs which help to increase the surface area so that the root can efficiently absorb more water and minerals. So this is about epidermis. Now coming to the second region. The second region, I told this is parenchymatous, right? And we said this is single layer, one layer. All these things are important. Now coming to the second portion, that's called cortex. The cortex of dicot root is differentiated into two parts. General cortex and the innermost region of cortex is called endodermis. Now when we have to talk about general cortex, it is several layered. Not one layered, it is several layered. And that is also made up of parenchymatous cells only, but loosely packed. Loosely packed parenchymatous cells we can find in cortex. Let's draw. So the cortex is several layered. So I'll draw one or two layers. So I'm showing so many gaps between. That's it. So this is general cortex. And coming to the innermost region of the cortex is called endodermis. Enda means in, exa means out. Endodermis. This endodermis is single layered. The endodermis is one layered and it is inner to the cortex. And these endodermis of root, in some regions, the radial walls, this is a radial wall, and the tangential walls, the radial walls and tangential walls, so are having depositions with water impermeable material called suberin. Not at all throughout the endodermis, in some regions, the radial walls and the tangential walls. The radial walls and tangential walls are deposited with a water impermeable material. What is that water impermeable material called? Suberin. That suberin prevents the leakage. And what is such a region called? This is a Casparian strip. And here is again one more Casparian strip. Casparian strip is continuous or discontinuous? It's discontinuous. It's here, it's not here. It is here, like that. So we are talking about endodermis. The endodermis is how many layered, children? It is one layered only, I said. And with cells, of course, it is parenchymatous cells. And how are the walls? The radial walls as well as the tangential walls are deposited with which substance? Water impermeable waxy substance called suberin. What does it do? It prevents water leakage. It prevents water leakage. And what is such an area called? Casparian strip. What is this area called? Casparian strip. So many times in PYQs, in previous NEET examinations, they have asked, the endodermis of root is called Casparian strip. What is the function? It prevents the water leakage. And how many layered? One layer. What depositions it has? Suberin depositions. Where? Radial walls and tangential walls. Now let's talk about the third region. The third region is called as stele. The third region is called as stele. Now stele has three parts. The outermost region of stele is called pericycle. Where is pericycle? It is inner to endodermis. And the pericycle is one layered or few layered? That is few layered, made up of parenchyma cells having thick walls. Okay. So the pericycle is few layered. So I need to take few layers of pericycle. So one layer is this. One more layer. One more layer. So they told the pericycle layers, there are few layers. So I just draw 3 layers. And the walls are thick and they are parenchymatous cells only. The walls are thick and they are parenchymatous cells. Okay. So what is pericycle? Pericycle is few layered in root and it is made up of thick walled parenchymatous cells. It's made up of thick walled parenchymatous cells. What are the functions of the pericycle in dicot root? So when we say the taproot branches right into lateral roots, the lateral roots originate from pericycle onwards. That's the first function of pericycle. What's the first function? Origin of lateral roots. Endogenous, from innermost part they are arising. From here the root will arise, right? That's lateral root. That's the first function. As I told, this is primary stem. During secondary growth, what happens? Vascular cambium and cork cambium are formed. So now from where the vascular cambium and cork cambium originates in dicot root means it's again from the pericycle only. So the next function we say origin of a part of vascular cambium. Origin of part of vascular cambium is from pericycle. And the third one, origin of cork cambium. Complete cork cambium we can say. Origin of complete cork cambium is from pericycle. That's it. These are the functions of pericycle. One layered or few layered. Parenchymatous cells. Thin walls or thick walls or thick walls. And where it is? Inner to the endodermis. It's the outermost region of the stele. And what are the functions? Origin of lateral roots, formation of vascular cambium, formation of cork cambium during secondary growth. Now let's talk about the vascular bundles. Just now we saw in the vascular tissue system, the root will have radial closed vascular bundles. Radial closed vascular bundles means xylem on different radii. So I am keeping xylem on different radii. In clock directions, if I have to say 3 o'clock, 6 o'clock, 12 o'clock, 3 o'clock, 6 o'clock, 9 o'clock directions. Xylem is on different radii. Protoxylem peripheral, metaxylem inside, protoxylem out, bigger xylem in. What is this condition called? EXARC condition, very important point. So xylem on different radii and phloem on different radii. Here comes the phloem patch. Xylem on different radii and phloem on different radii. So if xylem and phloem are on different radii, what is it called? It's called radial vascular bundle and not having cambium between. So closed vascular bundle. How many xylems and how many phloems did we draw? 4 xylems and 4 phloems. Tetrarch condition. Minimum of 2 diarch. Maximum of 4 only. Tetrarch condition. So that's about vascular bundle. So we need to write means vascular bundles. We say it is radial and it is closed. And the condition of xylem is XR condition. How many other? Di to tetrarch condition. And the innermost portion, this is the second part, and the innermost portion of stele is called as medulla or pith. For dicot root, the medulla or pith is very small, inconspicuous or either absent. So the medulla or pith is small, inconspicuous, means not clearly visible. So, and it's made up of parenchymatous cells. It's meant for storage. Or it's absent also, we can say. That's it, children. We finished anatomy of dicot root. We have taken sunflower plant as a reference plant. The 3 parts of the anatomy of dicot root are epidermis, the first part. Second one is cortex. And third one is stele. So epidermis, it's made up of parenchymatous cells, tightly packed, elongated cells without spaces. Since it's an underground part, no cuticle. Since it's not involved in exchange, it's not meant for exchange, no stomata. And since it's not meant for photosynthesis also, its function is protection, so no chloroplast. Now coming to this region, that is general cortex, a few layers, several layers of loosely packed parenchymatous cells. Now coming to the innermost region of cortex is called endodermis. Endodermis at certain regions, it has radial and tangential walls deposited with a water impermeable substance called suberin. Such a strip is called Casparian strip. Right? This is Casparian strip. And then coming to the steel has three parts. The outermost portion of steel is called pericycle, which is few layered, parenchyma, thick walled cells. Three functions: origin of lateral root, formation of vascular cambium and CORK cambium. Coming to the vascular bundles, radial, closed, XR condition, direct to tetrad condition. Coming to medulla orbit, it is absent or it's very less. Take a screenshot, then we will talk about anatomy of monocot root. Right? If we have to see the anatomy of monocot root, we can take a grass root or wheat root as an example to discuss. But in Ncert, they did not specify any name as such. But for DICOT, they specified sunflower. But for monocot children, they did not give any name. So, DICOT, if you know, monocot is very easy to understand. The differences lies here only. We can quickly finish off monocot root. Let's go. Now for monocot root, it's not sunflower. For monocot root, epidermis is going to be the same what we studied for DICOT. Epidermis is same what we studied for DICOT. The second one, cortex. Again the same what we studied for DICOT root. Third one, endodermis. Again same what we studied for DICOT. So three things are in common. Now the fourth one, pericycle. Three things are going to be different. Pericycle. What are we going to say pericycle? As I told, it is different in monocot root. So we all of us know that the monocots doesn't exhibit secondary growth. And if there is no secondary growth, vascular cambium will not be there. CORK cambium also will not be there. So pericycle, lateral root formation will be there. That point is common. What is that? Lateral root. Lateral roots originate from pericycle. That's common. But monocots exhibit secondary growth or no secondary growth? No secondary growth. So no secondary growth. If there is no secondary growth, there is no necessity of vascular cambium, no vascular cambium, and no CORK cambium also. So this is with pericycle. Now what's the next part? Vascular bundles. As I told, in root the vascular bundles are radial and closed. Means xylem on different radii, phloem on different radii. But the number of vascular bundles in monocot are many. The number of vascular bundles in monocot are many. So more than 6 Ncert has told. Since it is more than 6, so we can't say the number, we are just calling it as a polyar condition. How many xylems and phloems are there? Many. So they are more than 6. So then what is that condition called? Polyarc condition. Xylem on different radii and phloem on different radii still. So here is a phloem patch. Here again is a phloem patch. Here is again a phloem patch. Again a phloem patch. So xylem and phloem, too many are there. It's more than 6. So that's why it's called polyarc condition. Different radii, so radial vascular bundles, and no cambium between them, so it's called closed vascular bundle. XR condition still means protoxylem out, metaxylem in. Protoxylem out, metaxylem in. And in between xylem and phloem, there are parenchymatous cells to fill the gaps. Those parenchymatous cells which fill the gaps between xylem and phloem is called as conjunctive tissue. Now, what is that tissue which is present here, which is filling the gaps between xylem and phloem called? This is called conjunctive tissue. In DICOT also it will be there, in monocot also it will be there. So polyarc condition more than 6, this thing is common. What is that? Radial, closed. XR condition. And coming to the last part, that is pith or medulla. pith or medulla. It is prompt. It is more, means it is big, means it is conspicuous. Done. Monocot root, done with this. So 3 things are common, 3 things are different. Have a screenshot, then we will go to dicot stem. Now let's talk about dicot stem. Again, for dicot stem, we can take sunflower plant as a reference, or we can take cucurbita plant as also reference. Now in NCERT, for stem, they have given 2 pictures. Cross section they have given, and a small portion which is enlarged view also they have given. So we will see the cross section, transverse section of dicot stem. So, anatomy of dicot stem. We are going to take sunflower or cucurbita as a reference one. Now, as all of us know, the first portion is epidermis. For root epidermis, we use the word, do you remember? That's called epiblemma. But for stem, we don't give anything like that. So, epidermis, as all of us know, it is single layered. I'm going to draw a single layer, elongated parenchymatous cells without any spaces, compactly packed, without intercellular spaces. And all of us know that the stem has trichomes. The stem has epidermal appendages called trichomes. Trichomes are multicellular. They can be soft or stiff. They can be hairy or glabrous. They may be sometimes even secretory also. They reduce the water loss by transpiration. And since it is an aerial part, it has cuticle. Now this is cuticle. And it has few stomata. Leaf has many stomata. It has few stomata. Now coming to the epidermis, what should we write? 1 layer. Yes. And it has spaces or no spaces. It has chloroplast or no chloroplast. It has cuticle or yes, it has cuticle. And it has trichomes. It has trichomes and it has stomata. Few stomata. What can we write? I'm going to write few stomata. Now coming to the second portion, which is cortex. The cortex of dicot stem is well differentiated into 3 zones. The outermost region is called as hypodermis. The middle region is called as cortex or general cortex. The innermost region is going to be endodermis. The innermost region is going to be endodermis. The cortex of dicot stem is differentiated into 3 regions. The outermost region, which is below the epidermis, is called hypodermis. The hypodermal cells of dicot stem, it's few layered, and they are collenchymatous. Collenchymatous means corners are thickened. If corners are thickened, spaces will not be there, spaces will be occupied. I am just drawing 2 layers. This is going to be the hypodermis. The hypodermal cells of dicot stem are collenchymatous. Collenchymatous cells, the corners will be thickened. Wherever you find a corner, you're going to thicken with cellulose, hemicellulose and pectin. The corners are thickened. Since corners are thickened, gaps will be filled. So, compactly packed again you can use without spaces. And the function of this collenchyma, is to provide mechanical support. The function is to provide mechanical support. What is this region called? Hypodermis. How many layers are there? We're telling few layers. And which type of cells they are? They are collenchymatous cells. And what is the function of collenchyma? Mechanical strength. Mechanical strength, right? And spaces, no intercellular spaces. Now coming to general cortex. The general cortex cells, how many layers will be there? Few layered general cortex cells, loosely packed parenchymatous cells. I'm packing them loosely. Few layers of loosely packed parenchymatous cells. The general cortex consists of loosely packed parenchymatous cells. Loosely packed parenchymatous cells. How many layers? Few layers. Spaces, yes. Yes, spaces are there. And coming to endodermis. The innermost layer of cortex is endodermis. The endodermis of dicot stem stores starch. That's why it's called starch sheath. So here is the endodermis, single layer, compactly packed without spaces. The diagram itself is indicating it. It stores starch, so that's why it's called a starch sheath. It stores starch in the form of grains, starch grains. So it's called starch sheath. The endodermis of dicot root is called Casparian strip. The endodermis of dicot stem, it is called starch sheath. What is it called? Starch sheath, as it stores the starch. It is single layer, right? So this is about cortex. Coming to the innermost region, that is pericycle. We know that the pericycle also has three parts. The first one is going to be, it's stele. The stele is divided into three parts. The first one comes pericycle. Then comes vascular bundles. Then comes pith or medulla. Now coming to pericycle. The pericycle of dicot stem is this way. The pericycle of dicot stem. Now one point we need to mention here, that's about sunflower and cucurbita. Now if the hypodermis is so, pericycle is so, which manner? In sunflower, I draw sunflower only. In sunflower, I made hypodermis continuous. The hypodermis is collenchymatous and it is continuous. Now if we have to draw cucurbita, we have to make discontinuous. Here we have to draw and here we have to draw. In between we should not show collenchymatous hypodermis. So that means it is discontinuous. So in sunflower the hypodermis is continuous, whereas in cucurbita it is discontinuous. Then coming to pericycle, since the hypodermis is continuous, the pericycle is going to be discontinuous. The pericycle is discontinuous and is going to be sclerenchymatous. It is sclerenchymatous. Whereas in cucurbita, since the upper one is discontinuous, this becomes continuous. Now since I told we are drawing sunflower, we need to show it as a discontinuous one. And where do we find that discontinuous pericycle? Means on top of the vascular bundle. So for that first I should draw the vascular bundles. Now I told in dicot the vascular bundles are arranged like a ring. A ring shaped arrangement of vascular bundles is characteristic feature of dicot stem. So I am trying to draw the vascular bundles in a ring. So these are vascular bundles, and outer to vascular bundle is pericycle. The pericycle is in the form of patches, right? So it is discontinuous. It is on top of the vascular bundle. There is a semi-lunar patch-like structure. This is sclerenchymatous and it appears like a cap. So on top of the vascular bundle, so it's called bundle cap. The pericycle of dicot stem, that is sunflower, it is discontinuous. It is like a semi-lunar patch. You can see half moon shaped patches I am drawing. And pericycle is sclerenchymatous. This is collenchymatous, this is sclerenchymatous. It is covering the vascular bundle like a cap. Since it is called as bundle cap, right? This is bundle cap. If you have to draw cucurbita, make it continuous. I am drawing sunflower. Discontinuous. In NCERT, they mentioned about sunflower. What is this called? This is called bundle cap. That's about pericycle. Now coming to the next one, vascular bundles. So when we have to talk about vascular bundles. In vascular tissue system, we already told the vascular bundles of dicot stem, xylem and phloem are on the same radii, conjoined. And cambium is between xylem and phloem. So open vascular bundles. So let us show. The inner part is xylem. Xylem, xylem, xylem. Xylem is towards the inner side. and phloem towards the outer side. So xylem and phloem are on the same radii. So it is not radial, it is conjoint. This is phloem. And xylem and phloem are separated by cambium in between. So means it is open. This cambium between. So since cambium is there, it is open. Since they are together, conjoint. So conjoint open vascular bundles. So which type of vascular bundles are there? And protoxylem in, metaxylem out. ENDARC condition. So the vascular bundles, how many are there? Many vascular bundles are there. Each vascular bundle is top shaped or it is wedge shaped. How are they arranged? In a circular manner, in a ring like manner. That is a characteristic feature of dicot stem. Xylem and phloem are together. So it is called conjoint vascular bundles. And xylem and phloem are separated by cambium. So it is open vascular bundles. Protoxylem inside, metaxylem outside. So it is ENDARC condition. And the third one is medulla. So we can see there's so much of medulla or pith here. So when we have to talk about medulla or pith, it is there. It is big. And from the medulla, some parenchyma cells, real parenchyma cells, they radiate like this. They pass in between the vascular bundles to make primary medullary rays. So these are primary medullary rays. So they are originating from medulla and they are going in between the vascular bundles. What are these called? Medullary rays. So that's it about dicot stem. Now let's just see once again quick recap. So we can talk about sunflower or cucurbita. The outermost region is epidermis. It is single layered. It is having elongated cells, compactly packed without spaces. So it has epidermal appendages called trichomes. They are multicellular. They're soft or stiff. They can be hairy or glabrous, and they're even secretory. They can be branched or unbranched even. Now it has cuticle. It has fused stomata. It doesn't have any chloroplast. Now coming to cortex. The cortex of dicot stem is divided into three regions. The outermost region is called as hypodermis, which is just below the epidermis. The hypodermis of sunflower is continuous and it is collenchymatous without any spaces meant for mechanical strength. The second portion is cortex. The cortex is again few layered, loosely packed parenchymatous cells. The innermost portion of cortex is endodermis. The endodermis of dicot stem stores starch, so it's called a starch sheath. Now coming to the third portion, that is stele. The stele is also divided into three portions: pericycle, vascular bundles, and pith. The pericycle is discontinuous for sunflower. It is in the form of a semilunar patch on top of the vascular bundle and it is sclerenchymatous. Coming to vascular bundles, the vascular bundles are top shaped or wedge shaped. They are many in number and they are perfectly arranged like a ring, which is a characteristic feature of dicot stem. Xylem and phloem joined, so conjoint. They are separated by cambium, so it is open vascular bundle. Protoxylem in, metaxylem out, so ENDARC condition. Coming to medulla and pith, so much it is there, parenchymatous cells. And from the medulla, medullary rays will radiate in between the vascular bundles. They are called primary medullary rays. Take a screenshot, then we'll talk about monocot stem. Now let's talk about monocot stem. So, monocot stem doesn't have these many parts as we saw in the dicot. It's easy. But as we saw dicot root, monocot root, most of the points are similar. That many similarities are not there. Let's discuss monocot stem now. Now, in monocot stem, the outermost region is going to be epidermis. Epidermis is same, we can say, without stomata, but no trichomes. Only difference is trichomes will not be there. Now coming to the cortex. The cortex of dicot stem, we told it's well differentiated into three zones, right? Hypodermis, general cortex and endodermis. Here only hypodermis is there. In the cortex, what we see, only hypodermis. Let's draw. So, epidermis, single layer, elongated cells without spaces, parenchymatous cells, thin wall cells, meant for protection, little amount of cytoplasm, no trichomes. So this is about epidermis. Now coming to hypodermis, right? Just below the epidermis is hypodermis. The hypodermis of monocot stem is sclerenchymatous. It is sclerenchymatous. It is few layered and it is sclerenchymatous. The hypodermis of dicot stem, we said it is collenchymatous. But hypodermis of monocot stem is made up of a dead tissue. What's the name of the dead tissue? Sclerenchymatous. The function of sclerenchyma is also to provide mechanical strength only. So, it is a dead tissue. And what is it called? Sclerenchyma. The hypodermis is sclerenchymatous. And it's a dead tissue. Now, this is the second portion. Then scattered inside, we find vascular bundles. And what is that inside portion? We have to name it as ground tissue. Now the third portion, the hypodermis, is sclerenchymatous. And what is the function? Mechanical strength. And what is the whole part called? Ground tissue. And ground tissue is parenchymatous. So many layers are there, are completely occupied. Now, then comes the vascular bundles. Now, we say that scattered in the ground tissue are the vascular bundles. Now, the vascular bundles, the outer vascular bundles are small. So, there is no ring-shaped arrangement. The outer vascular bundles are small. They are near to each other and they are more in number. Whereas the inner vascular bundles are big and far from each other. They are arranged in two types. What are they? The vascular bundles, outer and inner. The outer vascular bundles are small. The inner vascular bundles are big. Outer vascular bundles are near to each other. Inner vascular bundles are far from each other. And outer vascular bundles are more in number. Inner vascular bundles are less in number. Now, if we have to see each vascular bundle, the vascular bundle is an oval-shaped structure covered by sclerenchymatous bundle sheath. This is sclerenchymatous bundle sheath. In dicot stem, we told the pericycle is like a patch, but here it is like a sheath. What is this? The vascular bundle is surrounded by sclerenchymatous bundle sheath, and they are oval vascular bundles, and inside the vascular bundle what should be there? Xylem and phloem. This patch is phloem and xylem is this side. Metaxylem out, protoxylem in. Since it is monocot, phloem parenchyma is absent. What is this? This is phloem and this is xylem. Which component of phloem is absent in monocots? Phloem parenchyma. Important point. Phloem parenchyma is absent for monocots. And xylem, how is the xylem? Xylem is in the shape of a Y. So these two are metaxylems and these two are protoxylems. So 4 xylem vessels are there, 2 metaxylems and 2 protoxylems. The protoxylems will sit one on top of each other and they crush each other. Since they are crushing off, they make a water containing cavity called schizolyserginous cavity. The protoxylems, what is this? The protoxylem sits one on top of each other and they crush each other. They crush each other and they make a water filled cavity, water containing cavity. 2023 need point. Water containing cavity. What is this water containing cavity called? Schizolyserginous cavity. It is called schizolyserginous cavity. So phloem is this side, xylem is inside, and metaxylem out, protoxylem in, means it is ENDARC condition. And xylem phloem together, children. Xylem phloem together, so it is conjoined. There's no cambium between them, so it is closed. Conjoined, closed, ENDARC condition. How many vascular bundles are there? Many vascular bundles. Outer ones are small, inner ones are big. Outer ones are near to each other, inner ones are far from each other. Outer ones are more in number, inner ones are less in number. Each vascular bundle is spherical to oval shaped structure. It's covered by sclerenchymatous bundle sheet. Phloem is like a patch, and xylem. Phloem parenchyma is absent for monocots. Then the xylem is arranged like a Y-shaped structure. These two are metaxylems, which are arms. The stalk is protoxylem. The protoxylem sit on one on top of each other and they crush off to make a water-containing cavity called schizolysogenous cavity. Right? Take a screenshot. Then we'll talk about dicot leaf. Now let's go to dicot leaf. So if you see the dicot leaves, on the upper side there will be dark green, lower side there will be light green. So they are not the same colors on both the sides. So it's called dorsiventral leaf. So dicot leaf is also called dorsiventral leaf. Monocot leaf will be called as isobilateral leaf. Now let's see. We're going to discuss the anatomy of dicot leaf, which is also called dorsiventral leaf. Right? So now, a leaf has two surfaces, this side and this side. So this side one epidermis and this side one epidermis. So the two epidermis are called as adaxial epidermis and abaxial epidermis. And leaf has stomata. Leaf has stomata, but dicot leaf has more stomata on the lower side only. So this epidermis is called adaxial epidermis or upper epidermis. Lower epidermis is called abaxial epidermis. I'm drawing the stomata. Right? So since it's an aerial part, it has cuticle also. It doesn't have any appendages, right? So this is cuticle which I'm showing. These are the guard cells. Right? So what is this part? This is upper epidermis, adaxial epidermis, cuticle. This is abaxial epidermis or lower epidermis. This is upper. It has stomata. And these two are guard cells. And we know the guard cells are being shaped. Inner to the guard cells, there is a substomatal cavity where air will be filled. This is the substomatal cavity. In dicots, the number of stomata are more on the lower side. So we call it as hypostomatous condition. They're either less or absent on the upper side. And the remaining portion is called as mesophyll. What is all the remaining part called? All the remaining part in the leaf is called mesophyll. The mesophyll contains mesophyll cells. They are parenchymatous cells, which are of two types. One type of parenchymatous cells which are pillar-like, which are compactly packed in 2 to 3 rows. So they are called palisade parenchyma. Now this type of parenchyma cells which are in 2 to 3 rows, they are pillar-like and they contain more chloroplasts. They are compactly packed. They are called palisade parenchyma, which are towards the adaxial side. Coming towards the abaxial side, in abaxial side they are loosely packed with air cavities. That parenchyma is called spongy parenchyma. So here also we are going to keep dots. Dots indicate chloroplast here. The number of chloroplasts on the adaxial side in the palisade parenchyma will be more, whereas in spongy parenchyma they will be less. So chloroplasts are more here. That's why it gives dark green color appearance for the upper side of the leaf. These are loosely packed, right? There are no spaces. Here there are too many spaces. What is this parenchyma called? Spongy parenchyma. And these are air cavities. This is spongy parenchyma, right? And these are air cavities. Now we need to talk about vascular bundles. So dicot leaf shows reticulate venation. The lamina, this is the midrib, and these are the primary veins, secondary veins, and veinlets and all. So the this thing, the veins are of different diameters. So inside the vein will be the vascular bundle. The vascular bundles are also of different sizes. In the midrib, the vascular bundle will be quite big. In the primary vein, little less. In the secondary tertiary veins, little less. The vascular bundles are of different sizes here. So the vascular bundles are spherical, and the vascular bundles are covered by parenchymatous bundle sheath. For dicot stem, you told it is sclerenchymatous bundle sheath. So point to be noted here, the vascular bundles of dicot leaf are covered by parenchymatous bundle sheath. And this is a vascular bundle. The vascular bundles are of different sizes. And where is xylem and where is phloem? So xylem is towards the adaxial side, phloem is towards the abaxial side. This is phloem, which is towards the abaxial side, and xylem will be towards the adaxial side. So xylem ada, phloem aba, and they are on the same vascular bundle, so conjoint. Leaf will never exhibit secondary growth, so means closed. So the vascular bundles are of variable sizes. It depends upon the diameter of vein. The vascular bundles are spherical. They are covered by parenchymatous bundle sheath. They are covered by parenchymatous bundle sheath. And the vascular bundles are conjoint, closed vascular bundles. Xylem is adaxial side and phloem is abaxial side. No cambium, so it is closed vascular bundle, right? So this is about anatomy of dicot leaf. So we are telling in dicot leaf, upper epidermis and lower epidermis. Upper epidermis also has cuticle, lower epidermis also has cuticle. So the cells are compactly packed and cuticle will reduce transpiration. The number of stomata in dicot leaf are more on the lower side. What is that condition called? This is substomatal cavity. This is substomatal cavity. More stomata on the lower side is called hypostomatous condition. And the remaining part is called as mesophyll. The mesophyll of dicot has two types of cells. The mesophyll of dicot has two types of cells. One is called as palisade parenchyma. The other one is going to be spongy parenchyma. This is palisade parenchyma, this is spongy parenchyma. Palisade parenchyma are elongated cells, compactly packed without spaces. Spongy parenchyma are oval shaped cells, loosely packed. That's it. Take a screenshot, then we'll talk about monocot leaf. Now the last topic in the chapter is monocot leaf. I told the monocot leaf is called as isobilateral leaf. So isobilateral leaf means both the sides, both the faces, it has the same color. Why so? Means it doesn't have two types of mesophyll cells. It has only single type of mesophyll cells which gives uniform color for both the sides. So the last topic is anatomy of monocot leaf, which is also called isobilateral leaf. Similarly, it also has upper and lower stomata. Isobilateral leaf will look like this, right? Grass leaf. So it has adaxial epidermis and abaxial epidermis. So let's draw. The thing is, the number of stomata are equal on both the sides. Amphistomatous condition. They are present on both the sides. Almost they are equal on both the sides. So one stomata at that side I draw, one stomata this side. Since it's an aerial part, it has cuticle. The monocot stomata, the guard cells are dumbbell shaped. So this epidermis is adaxial. Upper epidermis. Cuticle. Stomata. This is again cuticle. abaxial epidermis. Stomata. Stomata equal on both the sides. That condition is called amphistomatous condition. The middle portion is going to be called as mesophyll. In leaf, the ground tissue is mesophyll. The monocot leaf, the mesophyll cells are not differentiated into two types. They are same type. So they are they're just called parenchyma cells. We'll just call them as parenchyma cells. With chloroplasts, which are photosynthetic centers. Monocot leaf shows parallel venation. Parallel venation means all the veins are of same size, then the vascular bundles inside should also be of same size. So these dots indicate chloroplast. Inside the chloroplast, photosynthesis will be done. Chlorophyll pigment will be there. So one type of mesophyll cells only I showed. They are parenchymatous, they have chloroplast, they do photosynthesis. They are just called mesophyll cells. And monocot show which venation? Parallel venation. Parallel venation means all veins are of same diameter. They are of same diameter. So if all veins are of same diameter, then all the vascular bundles are of same size. Except the midrib vascular bundle, which is slightly big. Except the midrib vascular bundle, which will be slightly bigger. Right? And the vascular bundle is same. It has a vascular bundle surrounded by bundle sheath, and in leaf the vascular bundles are covered by parenchymatous bundle sheath. So this is parenchymatous bundle sheath, and the vascular bundles are conjoined closed, and xylem towards the eda, phloem towards the aba. This is phloem and xylem is here, eda. Xylem is eda, phloem is aba, no cambium, so closed vascular bundles, right? And for support, they have sclerenchymatous patches on both the sides. For support, they have sclerenchymatous patches on both the sides. So in NCERT, they showed us. So on both the sides, it has sclerenchymatous support. This is called as sclerenchymatous patch. So to summarize monocot leaf, it is called isobilateral leaf. It contains adaxial epidermis and abaxial epidermis. Mesophyll cells are not distinguished. They are of same type. They have chloroplast. The vascular bundles are of same size since the venation in monocot leaf is parallel venation. Here one point. So on the adaxial side, along the veins, there are some special cells which are empty, which are colorless cells, which are called as motor cells or bulliform cells. They are empty, colorless, large cells. Where are they present? On the upper epidermis. Along the margins, on the veins, they are present. Those cells are called motor cells or bulliform cells. Early morning, when moisture is there, so these cells will be filled. That means they become turgid. When they become turgid, now they expose themselves, right? So early morning, they help in unrolling. They expand. Whereas afternoon, when moisture is not there, it is too sunny, the cells will become empty, then they become flaccid, then they are shrunken, then so then they help in rolling. They roll towards the inner side. They roll towards the inner side. What are those cells called? Motor cells or bulliform cells. This is a PYQ question. Take a screenshot. Hope you understood. This chapter is important in terms of NEET. We'll meet soon. Thank you.